So let's start. First question.
Ma'am, you showed us an image where glucose is almost all the time is conserved highly in brain.
Cancer cells, yes.
In brain, they are in normal condition too.
Yeah, so brain eats up a lot of glucose. So if you do PET scanning, brain will always light up, okay? Because glucose consumption is highest in the brain. So when they are looking for neurodegenerative diseases, they do glucose scanning, PET scanning, and then actually neurodegenerative diseases, glucose consumption is a lot lower. So that is how they know there is something wrong in the brain.
So how do they identify the brain cancer, the brain tumors, if it's always high on glucose?
I think there has to be some high means high, but in cancer probably it's even more higher. So if it is homogeneously high, probably it is not that high, but if some part is really lighting up, then with contrast you can see that it is, very high. Also, when you give radioactive thing, bladder will show up because a lot of radioactivity is leaching from there. Then sometimes kidneys will show up, not because kidney has cancer, but it is trying to get rid of it. So many, bless you, many organs that will show, many organs will show like that. So people who do this kind of reading, they are used to it. So, but the image I showed you was extremely nice, like how bad was the metastatic cancer at just one targeted therapy. A person having that kind of metastatic cancer is just really bad to start with, and then one therapy is doing such wonders. So, just VRAP, just one VRAP.
They are using a certain kind of fluorescent
Oh, you should use this so that because go ahead. What is the question?
No, I'm answering to his question. Like I have seen in case of brain tumor cells detecting, what they do, they give IV which is having some sort of fluorescent compounds, and in case of a tumor cell in brain, that fluorescent compounds just light up there only at the tumor part only.
So if
and they perform the surgery and all.
So what happened? No, so for that they have to know exactly what is wrong with that one. So then they can give it. But the good news is that they don't have to do biopsy. They can try different things and they will know what is lighting up. So that is also one way of finding out what is overexpressed, right? And then like folate receptor and things like that, there are some which are generically overexpressed. So whatever is generic overexpressed in brain cells, you can give those also.
Are the neogenic?
Give it to, yeah.
So, are neoantigens unique to each individual, or there are some neoantigens that are common in every tumor? Like in general, any person have tumor, they will have this kind of neoantigen.
I'm sure there are some common ones and there are some unique ones. So not every tumor will have all unique. There may be hundreds of neoantigens, right? Hundreds of neoantigens. So some will be tumor specific and many of them will be common among tumor cells. Definitely. See, think about it in this way that all the cells in the body has the same DNA, right? Every cell has the same DNA. Even then, all different cells look different, like neurons look different. So a vast majority of the proteins are the same in all cells, right? Because they are the housekeeping thing, they keep the cell alive. And then they are unique proteins which give their unique structures. So in tumors, a lot of mechanisms are the same between different tumors. So those, and which do not exist in normal cells. So those kind of things are going to be shared between different tumors. Or you can say all blood cancers will resemble each other more than solid tumors. right? And then there will be obviously tissue-specific antigens. So for CAR T therapy, just one second, the challenge is to find the real right antigen so that it can target there. And the good news is that it's one-time infusion. Once you give the infusion, it stays there for years because they keep on proliferating in the body. But then I did not tell you about T cell exhaustion. So now scientists are working on why T cells are getting tired inside the body and how you can keep them alive, like they don't get tired. That's called T cell exhaustion. Go ahead.
Ma'am, like what mechanism do cancerous cells like use? Like they are like too efficient in terms of mutations. Like they
In
are
terms of mutations.
yes, like they are keep on mutating. And is this mutations are like randomly, like they keep on mutating, and some mutations are like randomly selected, and these mutations are like the better one survival for them, and they like keep on surviving, or like is it something that they are really too smart so that they can mutate in a way only so that they can restrict the body cells only?
Yeah, so the way so suppose you give Gleevec, I'm giving you a real life example, right? So Gleevec binds in a certain way. So let's say Gleevec has a side chain which binds here, okay? And the residue is touching is this one. So then what they will do, to avoid Gleevec, they will mutate this residue. So they are very smart to know which one to there. So if they add a large hydrophobic amino acid here, Gleevec will no longer bind. Do you see? So there was a threonine there, 315 threonine, which was converted to isoleucine. Isoleucine side chain is bigger than threonine. So cancer cells are not completely randomly doing it. They know that Gleevec is touching here. So if they replace threonine with isoleucine, that will interfere with Gleevec binding, and it became resistant. So they are not completely randomly doing, they know exactly which residue to mutate. There may be some permutation where they change threonine to isoleucine, leucine, few others. Those ones survived which didn't bind Gleevec. If they were still binding Gleevec, they will die anyway. So that's how the selection is occurring. But cancer cells are smart that they are not randomly mutating the whole pocket. They are also mutating the whole pocket and elsewise. So some mutations are going on, but they can be extremely precise. So when antibodies are made, right, against any antigen, the way we understand is that trillions of antibody molecules are made, and then the ones which survive are the best ones, right, which are the binders. I don't think tumor cells go to that extent of making those many mutations. They are obviously making many mutations, and it is easy. If you bind Gleevec, you die. We don't need you. The ones which do not bind the drug, they are the ones which survive. So it's automatic selection. So it's midway. They are very smart, but they don't know exactly which mutation to make. So they're making more than one. And only the survivors survive. So these have high, like, one of the ways they survive is they are undergoing mutation. They are expressing MSC1, then they are not expressing MSC1. They are exposed to something, they do something else. So they are constantly evolving, just like virus. You develop a drug against one virus, the new virus comes in. Cold medicine, every year we get the flu shot, right? Because a new variant comes and the new variant comes and the new variant comes. So cancer cells in that respect are like that. Otherwise there will be no resistance. Resistance emerging in 3 months tells you that they are mutating fast. Normal cells don't mutate because they are afraid of turning cancerous. So they are happy the way they are. Only they are exposed to extreme conditions like UV radiation or something, then they will mutate. Cancer cells are inherently too plastic, changing all the time. But if you don't apply any pressure, I don't think they will undergo so much mutation. It is under pressure that they mutate. So when you give chemotherapy, they become very resistant. That's when they're mutating to survive. Right? Go ahead.
Ma'am, not every cancerous growth's fate is to metastasize, right? Some can be localized and stay that way for the.
That is true.
So, ma'am, is there a checkpoint that the cells cross after which they are like they have to do the EMT and have to move away, or is it completely randomized?
No, it is not randomized. So it depends on the kind of cancer also. Some cancers are inherently more aggressive, others are not. Even in the same tissue type, some cancers are aggressive, others are not. So breast cancer is a good example. It has 3 receptors: estrogen receptor, progesterone receptor, and the third one is I think EGFR receptor or something, HER2. So if all these receptors are gone, okay, then the cancer is aggressive from the start. Okay, similarly in pancreatic cancer or hepatocellular cancer, they all have different cancer type. Some are more aggressive than the other. So those cells divide, they are more motile, they are more aggressive, they metastasize. So yeah, it is quality of the cancer, which cells are those and how they are mutating.
Ma'am, so would it be right to say that if you are applying more pressure, there is more odds of metastasis happening, or is it just aggression that is determined by the nature of the cancer that decides what's happening?
If you are applying more pressure, the chances are they will die also. But if they are survivors, the survivors will be more resilient, right? But you have to apply a lot of pressure. You have to do everything to kill the cancer cells. But if you're successful in killing it, it is gone. But if you're not successful and they survive, it metastasizes somewhere else. But the thing is this, that we know a lot of people whom we don't know, we just read about them in news. They survive, right? They survive for many, 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 many years. So we have made a lot of progress.
Ma'am, you told about that a combination therapy like PDL1, PD1 and CTLA4. Like what exactly is happening in that combination therapy?
So in that combination therapy, so there are 2 things which are occurring. So cancer cells are upregulating immune checkpoint things to prevent to shut down the immune system. So in some patient PD-L1, PD-1 is occurring, and some cases CTLA4. The chances are both are upregulated in patients because the tumor is growing, right? So it is an approved therapy to give PD-L1 along with CTLA4. And the oncologist at Max has said that in his experience, when you give PD-L1 along with CTLA4, that is better than giving PD-L1 alone with VEGF inhibitor. But my thinking is that anti-angiogenesis inhibitor, this is not documented in any paper, any journal. This is his personal experience. But in my opinion, angiogenesis inhibitor is very important. So I would prefer PD-L1 angiogenesis inhibitor. I have not seen CTLA4 angiogenesis inhibitor, but that is also possible. But most of the time, if you have to give one immunotherapy, PD-L1 is the choice one. So I think that is more commonly changed. But people also, if you check online, if you give PD-L1 and CTLA4 together, that's more toxic to the patient also. It has more side effects. So if the cancer is very aggressive, that's when they give that. You know, in my personal opinion, I have seen all the people who are taking immunotherapy, they're getting better. but in literature it says only 20% people respond, and I don't think so. So it may be in that particular study 20% people responded. It also depends on the kind of cancer. Immunotherapy is fantastic for blood cancer. And also, there could be other factors also. Person's lifestyle and all those things could contribute to that. There could be other comorbidities there. So every person is different. And I don't think people have done PGX along with immunotherapy. So if they do PGX also, maybe they will find something different from patient to patient to patient.
Okay, ma'am. So my question is coming from APC and T cell interaction, and that too from the second signal, where B7 binds with CD28, and for that inhibition, there's a CTLA4. So what cancer cell do is just the overexpression so that this interaction gets inhibited.
So cancer cells themselves are not expressing it, they are recruiting Treg cells to do that.
Right.
That's the difference, yeah.
Right. So we are using inhibitors of CTLA4.
Yes.
Just in this contrast, if we're using inhibitors, so aren't we blocking CTLA4 will not create T cell hyperactivity?
At that time, you really want to target the tumor cells. So that time hyperactivity is a lesser evil than getting T cells activated, because it is such a immunosuppressive environment that activating the immune system is more important. That's why there is this toxicity issue. Whenever you are giving immunotherapy, there is always a chance of autoimmunity, always a chance of autoimmunity. So this is, doctors tell you in advance, there could be autoimmunity there.
Just to overcome this autoimmunity, do they have different mechanism to counteract this? Because our body is in constant haphazard to manage all the toxicities and all those stuff.
There could be a lot of different mechanisms, right? But quietening the T cell itself is a very important thing. So if you're activating it, I would imagine there is a chance of some autoimmunity. But then also it depends on the dose, right? It also depends on the dose. And then I don't know. So in general, what happens is that T cells, they express CTLA4 only when they themselves think that they need to be quietened. So initially, CTLA4 is not present in T cells. They have CD28 and they have obviously TCR, right? But when they are activated, then that's when CTLA4 is overexpressed to shut down this. So it's like they are themselves putting CTLA4 so that the signal is quietened, right? So I don't know if CTLA4 is mainly present at the tumor site because it is such an immunosuppressive environment. So then it becomes more specific there, right? Because I don't know how many T cells have CTLA4, because once they are activated, that's when they express CTLA4. So if tumor cells are, if T cells are not even activated anywhere in the body, why would they even bind that, right? Because if they were inherently expressing CTLA4, then there will be no T cell activation ever. So only when they are primed already, then they express CTLA4. So now you can imagine that the tumor cells, Tregs, are the ones which are expressing more CTLA4. So the therapy is much better there. And if it has metastasized to different parts of the body, it will be immunosuppressive environment there. So they will work at all those places. I have seen that, tumors which have metastasized as other part of the body, I've seen the tumor disappearing from those places with immunotherapy. Small things disappear. So it is a good one. It is a good one. But looking at CAR-T, it is really good one. CAR-T is really good one.
Ma'am, so as I do daily a question about the oncolytic viruses, my question was that, ma'am, if we are to, what the mechanism of oncolytic viruses is that those will latch on to the tumor cells, insert their DNA, and then ultimately they will proliferate and lyse the tumor cell and expose the antigens to the immune system. So my question is that when the replication process will be happening, the DNA replication of the viral, whatever designed genome that we have put in, it will be happening inside the cell, which is an environment with high oxidative stress and all. So won't those replications also be erroneous in some sense? Or would those be conserved?
Viruses, they don't care. Viruses survive because they mutate, right?
Ma'am, but we have put specific things with an intention of their action, right? So what if what we programmed ends up getting modified and the effectiveness of the virus ends up going down?
I don't think so. Virus will only mutate if it is not able to replicate. If it is able to replicate, it won't change itself. So even if some viruses are mutating, you will have plenty of virus. You have not worked with virus. So what happens is that when you do virus infection, trillions of molecules come out of the cells. So even if some percentage is mutated, it will not make any difference. Survival of the fittest, the guys which are infecting, they are the ones which are going to survive. The guys which are not able to infect will be gone. So it is going to work for our advantage there. And somebody asked me yesterday about the autoimmune thing, so I checked today. They do not affect normal cells. The autoimmunity is pretty low, like it's almost negligible there. So we are very good in designing that it only replicates in tumor cells, not in normal cells. So it showed in pictures again and again, normal cells are unharmed. So that seemed like a very cheap option to get rid of tumor cells, right? And you don't even need antigen because you are infecting a virus and virus itself is clean. Like we do viral infection all the time in our lab. So there are Bosc cells and Phoenix cells which are used to make virus. So when you harvest the virus, the cells actually lyse because cells are so sick, right? They are good for nothing after that. They just pop up and release the virus. So the cells are completely gone. So tumor cells are gone. And imagine the virus is infecting. So then the good news is that it cannot even mutate to fix the virus because virus will outbeat them, right? So that seemed like a very good one. So I don't know why it is not used widely. That is something I'm going to check it today, exactly what is going on there. Tomorrow we'll study stem cells. Okay, why we study stem cells is that there are many reasons to study stem cells. With respect to cancer, cancer many times leads to damage to tissue because of radiation, because of surgery. So you can use stem cells to regenerate those things, and that's why stem cells are very important for cancer also. But stem cells are in general very useful. Have you guys studied stem cells?
I've learned about hematopoietic stem cells.
Hematopoietic stem cells, yeah. Have you guys studied iPSC cells? No. Induced pluripotent cells, yeah. Yeah. We'll see all of those tomorrow. All right, guys, next question. Yes, Vanshika.
You told us that when BRCA1 and BRCA2 gets mutated, PARP will perform their function like double-stranded DNA break repair. So, and that is a tough repair compared to single-stranded. So, is it efficient? Like, won't there be any mutations or like mistakes, errors?
There could be some mistakes and errors, but those cells get eliminated, but those which are fixed, they take over. So what happens is that if there is a mistake, those cells will die. But if PARP is able to fix it properly, those cells survive and they take over. So by any chance, if it is incomplete repair, then that cell will die. And the other cells which are fixed properly, they are the one which will take over. So most of the cells will be all right. Yeah. So cells have this bypass mechanism. So, I will tell you something. So suppose you have to, in our lab also we have seen, and everyone has seen, that if you want to, let me say it this way. Suppose there is a protein A, okay? So you have two ways to find the function. Either you delete that protein A using RNAi, which is a piece of cake, or you add a small molecule to inhibit it, or you add a PROTAC to degrade it, right? So when you do RNAi, so RNAi is relatively new, okay? I mean, it was discovered in late 90s, in 1990s, and then it became a general thing in the lab around 2003 and 4. 5, 6, it became a regular process, right? Before that, it used to take a very long time to get rid of a gene, okay? But even if you use RNAi, it will take 24 to 48 hours to get the protein expression down, right? So 24 to 48 hours is a long time for cells to bypass that signaling. So what if you remove that protein? The cells will use some compensatory pathway to survive. So some cells will die if it is an important protein, obviously, but some cells, however few cells, will find a compensatory pathway and they will survive. So you really don't know the function of your protein because the cell survived, and you think it is not important for survival because you knocked it down and the cells are surviving, right? But if you use a small molecule inhibitor and inhibit it, it is so quick, you add it 5 minutes later, it is gone, and then the cells will die. And then you know, yeah, this protein is really involved because you did not give the cells time to compensate. Like you did not give them 24, 48 hours. When you're using RNAi, they're going slowly down. 100 to 95 to 90 to 85 to 80, slowly over a period of 24 to 48 hours. So that is plenty of time to for cells to think of an alternate mechanism. Even if they're not cancer cells, they are living cells, right? That's how we live. We are exposed to one adverse condition, we adapt and we adapt and we adapt, right? So, but if you give a small molecule, it is an instantaneous effect. So then you will see the real picture. So we expressed a protein which is supposed to cause oxidative stress. We didn't see oxidative stress because it takes time to express the protein. But when we added the protein directly to get it across the cell, there is a way to do that. Instant oxidative stress, because they did get chance to. When we expressed it, the antioxidant proteins were upregulated, so we didn't see oxidative stress. But when we added, it was so quick, 5-10 minutes later, oxidative stress increased. So the point I'm trying to make is that cells are pretty resilient. So when you use small molecule versus long process, they will give you oftentimes different results. That is why people use a combination of things, small molecule versus this versus this. Next question, please.
Ma'am, my question was, when BRCA1 and BRCA2 are not present in the cell, we give PARP inhibitors to destroy their cell repair mechanism, so they will die eventually. So ma'am, if the mutation for BRCA1 and BRCA2 is not sporadic but germline, and every cell of the body does not possess PARP, then it would kill the person.
That is a good point. Because every cell will have BRCA1, BRCA2 mutation if it is a germline mutation. And if you give PARP inhibitor, it should affect all the cells. So here is my answer to that. obviously people survive. It's a very good drug. So I think normal cells have alternate mechanisms to take over it. Cancer cells are more more vulnerable to that. But let me check that. That's a very good point. Actually, when I was teaching you, for a second this thing came to me, but before I could grab that thing, because I was talking, it slipped off my mind, because I was thinking it is in every cell of the body, but by that time I was speaking something else, so I forgot that I was thinking. But let me check on that. That's a very good point. That is a very good point. But it is, in general, it is known that normal cells are really efficient in DNA repair because they don't divide as much. So they could have some additional mechanisms there which allow them to survive. Cancer cells are dividing so fast, and that is the backbone of chemotherapy. That because they have defect, even if BRCA1, BRCA2 are there, DNA repair pathway is defective in cancer cells. So they are more prone to damage, and that's why they are more prone to. So, yeah, okay, I will answer your question. Now I remember what's going on. So, when you give olaparib or something, you're also introducing DNA damage there by radiation or chemotherapy or something. Otherwise, under normal circumstances, why would they die? So you are introducing DNA damage. So it is often given in combination with some kind of therapy which causes DNA damage.
Told us that in normal scenario too, our DNA is damaged daily in every cell, and it is repaired on daily basis.
That is true. That is true. But there are different kinds of DNA damages, right? So cancer cells are more prone to danger, like double-stranded breaks are very bad. So double-stranded breaks occur when we are exposed to UV radiation, like serious UV radiation or some kind of serious chemical. What happens in daily basis are like deep urination, deep pyrimidation. So it's like, the gums are there, so the gums are phosphodiester linkage, so that is intact, but the tooth is missing, so the base has gone. So they are very easy to fix. Cut it from either side, remove it, and add the correct base, seal it. Okay, if there are some other bases, you cut 12-13 base pair, cut it, fill it with DNA polymerase, ligate with ligase. So these kind of things are piece of cake. When there is double-stranded break, that's where it gets a little bit dangerous. And double-stranded breaks are not happening spontaneously unless you expose yourself to something bad. So if you are sitting in sun for very long, like people do, and sit in beaches, so we may not experience too much damage because our skin is dark, so we have more melanin which protects us. But people who are very white, they lack melanin, and if they sit in the sun, they are more likely to develop skin cancer, right? So these people, so those kind of damages will break both strands of the DNA. So a lot of DNA repair enzymes are mutated in skin cancer, lot of it. So you will be surprised how many cancers are caused by defect in DNA damage, DNA repair. So you can also imagine that it is possible for olaparib, and again I will check it today and then I'll tell you with like really confirm it tomorrow. But it is also, I have seen that they give localized some kind of therapy to introduce DNA damage and then give olaparib, and then those cells will die. Right? Makes sense? Yeah. But that's a very good point. It is in every cell of our body, no doubt about it. But think about chemotherapy, right? Chemotherapy is targeting every cell of our body. We are just hoping cancer cells are more susceptible. So there are bad therapies. I did my part, right, with 4 We'd finished before lunch. Actually, it was easy because you guys had studied a lot of it. Tomorrow it will be all brand new stuff because we have not studied stem cells.
Ma'am, in CAR-T therapy, you have discussed about the transvection or the transvectants. What are these exactly, transvectants?
Oh, so what happened? So tell me, has anyone here worked with bacteria and made proteins in bacteria? Nobody. No one.
I have done cell culture basically, but I haven't.
Okay. So it doesn't matter. It really doesn't matter. You guys look very apologetic and very this thing. Don't worry about it. I don't know the background, so I have to ask to start where I should start, right? So never be apologetic about it, okay? Whatever you are doing is great, is the greatest what you are doing, so never feel that way, okay? So what happens is that, so let's start with bacteria, right? So do you guys know what a plasmid is? All of you know? Is there anyone who doesn't know plasmid? All of you know, right? You know Evans too, right? Plasmid. Okay, so there are bacterial plasmids and there are eukaryotic plasmids. So what that means is that DNA replicates. So when DNA replicates, DNA polymerase recognizes the origin site, binds there, and then it replicates the DNA and it stops at termination site. So E. coli has different, eukaryotic have different, that's why we have different plasmids. Makes sense? So E. coli is a very good machine to make proteins, human proteins, right? So we take a plasmid and we put human DNA sequence there, right? So, and then we feed it to E. coli. So how do we feed it to E. coli? So there are many ways. You can give them electrical shock, you can buy special cells, E. coli cells. So they are made in such a way that if you get them heat shock, so you put them on ice for 30 minutes, then you take it out and for 45 seconds you put them at 42 degrees or 37 degrees, and then you immediately after 45 seconds you put it on ice for 2 minutes. So they will get heat shock and they will take the DNA inside. So you mix your DNA of interest and you mix it with E. coli cells, not growing cells, these are frozen cells. So you buy these frozen cells or you can make competent in your cells. These are called competent cells. These are nothing but E. coli cells ready to take the DNA inside. So, or you can, it is very easy, they take the DNA inside. Then what you do, E. coli will drop the plasmid. So you put a selectable marker, so those E. coli who have taken will survive in ampicillin. Those who have not taken will not survive in ampicillin. So E. coli is forced to keep the plasmid inside. Now this plasmid encodes your gene of interest, let's say Sark protein. So this will keep on making Sark for you and you can purify it from E. coli. So E. coli is a protein production factory for us, for all of us, right? So that process, when E. coli or any prokaryotic cells takes DNA inside, it is called transformation. What is it called? Transformation. Now you can do the same with mammalian cells. You have done cell culture. So mammalian cells will not take DNA if you just add it and you get heat shock. There are reagents you can buy like lipofectamine and there are many of them. So you mix them, put on ice for 30 minutes, then you spread them, then you wait for 1 hour, then you wash and 24 hours later they will express the protein. So when you do that, that process is called transfection. Okay, so what happens is that the cells will take the DNA, but very few cells will take the DNA. Some cells like to take the DNA inside, so the transfection efficiency can be as high as 90%, but most of the cells will take 10 to 20%. So you will have protein expression of your gene of interest, but not too much. So then also you do selection there. So your plasmid has some selectable marker like puromycin or something. So if you grow the cells in that for a long time, only those cells will survive which have your DNA, right? So you can make a stable cell line. So then there is another way that you take your DNA of interest in a viral vector, and then you put it in a virus producing cell line, not your cell line, but a virus producing cell line. So because it is in a viral plasmid, it will make viruses, and your DNA is inside the virus. Then you take that virus and you add it to your cell. That process is called infection, not transfection. So now 100% cells will take the virus in, and you will get huge amount of protein, and your viral vector also has a selectable marker, so you can make stable cell line. So whenever CAR T therapy is happening, they are using viral thing. So in our lab, we never do transfection. From the very first day, we are doing infection because it is so efficient. You would never do transfection. So that's the difference between transformation. Transformation is in E. coli. Transfection is adding DNA to mammalian cells using some transfection reagent. And then if you make virus of your DNA, encoding your DNA, and then you add it to mammalian cell, that is called infection, not transfection. So that's the difference. So anytime these people are making, they are using viral vectors. So what you do, you take out T cells from the patient. Then for the DNA, you take the DNA for the CAR T cell receptor, and then you package it in a virus, you add it to T cells. Now these are human T cells, right? DNA will never go via transfection. Okay, so you have to use the virus, and then once they become stable there, then you re-inject it to the patient. That's what it is. So I showed you first generation, second generation, third generation. They are just making it more and more and more potent so that there is a more stronger reaction, like weaponizing it even more, right?
Ma'am, my question's coming from CAR T cell therapy again. So in a process, we isolate T cells from the patient, we treat them, we either modify them, or in this in between, do we expose them to some sort of antigen during this so that they get modified?
No. We already know the antigen before, so we have the DNA which makes the antigen in the same form. So let me tell you something. So what happens is that for nanobodies, for example, right? I will just tell you, even for antibodies. So there is no way that you and I can predict what DNA to make and what, because when antibodies are actually made in human body or in mouse or in rabbit or in camelid, trillions of molecules are made, and then nature selects for the best binder, right? So that we cannot predict. Trillions we cannot make, we cannot predict. So what we do is that first we make it in an organism, whether it's a mouse or a rabbit or camelid or something. Then we take that antibody and then we sequence the antibody. Okay. So we get the DNA sequence. Now we have the vectors, and we have in our labs too. So you can actually produce heavy chain and light chain together, express it in mammalian cells. And once the antibody proteins are made, they will automatically form the proper antibody. Okay, and then you can lyse them up, and there are beads which will bind to heavy and light chains, and you can isolate the antibodies and you can purify them. Okay, so now what you do, that you take a tumor antigen and you raise an antibody in whichever place you want to, like let's say mouse. Okay, you raise it in mouse. Okay, now you know then you know the sequence of the antibody. Okay, once you know the sequence of the antibody, then you make it in the lab. So now you know what part will make the heavy chain, what part will make the light chain. So now you keep on mutating it to make it humanized. So you don't change the variable region which binds the antigen, but you change the rest of the thing, right? And then once you do change, that is what humanizing means. You just change it. You are just not touching the variable region. And then that is, you make it in E. coli or some other organism. So this is recombinant DNA. You see, it's not coming out directly from the organism. You're making it in E. coli or some other organism. There are SF9 cells, insect cells, you make these. And once you make them, you purify them, ready to use it. So basically it is coming out of the DNA sequence. Once the nature selects for you, you find out what nature did, and then you make it in the lab.
Okay.
So you guys have heard of AdGene, I'm assuming. AdGene sells a lot of DNA. It is the biggest source of DNA around the world, and it's very cheap, ADDGENE. When I was at ISER Mohali, who is from ISER Mohali? You, right? So I was there in 2016, and I met, there was a lady I met. Of course, I can't remember her name now. I was in touch with her for some time also. So she was getting all the things from AdGene, so it is the biggest source of all the DNAs, and it's very cheap. It's not profit kind of thing. So it is like I have given some DNAs to AdGene, so for no cost. So it is like sharing, but they charge you some money because they have to, keep the whole organization going. So it is super cheap. It is just non-profit thing. So the thing is this, that so they sell a lot of antibodies thing. You can make it in the lab. It will assemble it in the mammalian cells or insect cells, and you lyse it up. You can purify it. Yes.
Ma'am, as you said, the neoantigens, there are some neoantigens which are common in all kinds of tumors. So why don't we make a vaccine based on those neoantigens and vaccinate every person? So as soon as a cell mutates towards a tumor, as soon as it grows that antigen, the immune system will
So
eradicate
this is
it.
going to come tomorrow, probably tomorrow, because tomorrow is the second last lecture of the series, right? So there is a topic called immunoprevention which will come tomorrow. So for a lot of things we know already, like HPV virus infects and leads to liver cancer. So you can vaccinate in advance such people so that they are not exposed to liver cancer. So for many viral induced cancer, there are vaccines available. Then they do genetic testing of many people who are more prone to get cancer. So they are also vaccinated against, for which things are known. So it is becoming a common occurrence now that one can get vaccinated for certain kinds of cancer. So our knowledge is not as yet at that we know the new antigen of everything right now. And keep in mind, not every new antigen will really work. You have to find the best guys which are likely to be exposed, right? When the antigen presenting cells are presenting, what they are likely to present. So the way they work is that they take a protein of interest or any protein, they make 8 to 13 mer, right? And then they expose. So not everything will have an immune response. So we have to really figure out what kind of antigen we have to use, which will trigger an immune response. So basically to make a vaccine, we have to know exactly what we have to use there. So for some, we know what kind of vaccine to make. For others, we don't really know what kind of vaccine to make. Is it making sense? So immunoprevention is a very big thing. So there are vaccines, preventive vaccines. Evans, question?
Okay, my question is about the surface molecules in the cells. I see
Which molecules?
surface structures.
Molecules in the cell?
Yeah, MHC, the CD, and many
28
others.
and all
Sometimes
those
this is
things.
really confusing to
Yeah, of
to
course.
know
It
which
is
one
confusing
is which.
to me
Yeah.
too. So many
Yeah.
of them.
So
Yeah.
many of them. So, yeah. So I struggle with that. Knowing CD, like MHC1 binds with which, like MHC8, no, MHC1, then T cell. CD4, yeah, those ones. I struggle with that. Okay, and my other question is about the viral. This is different from the cancer and inflammation. When they make vaccine, a virus vaccine, when it reaches the clinical levels, how do you test that? Because already you've injected the vaccine to the patient, and you need to see whether it is working in real disease. So for viral infection, how do you test? Because if you inject patients with, say, HPV or any other virus, you risk like infecting the patient with the virus.
Okay, so I will tell you that. So I will know exactly how it is tested also, because I met a person where it was tested. So he was working in his garden, and he was, taking care of the weeds and everything, like, we all have to do ourselves. So he got exposed to some mosquito or some insect, not mosquito, some insect. So he had a very, very strong reaction. It is a very famous thing. I can't remember. It is very common in South Africa also. There is a kind of bee or something, bee sting or something. Like, he had very, very strong reaction. So he went to the hospital, got tested. So they checked for the antibodies towards that. And he had the antibodies towards that because the body's immune system was reacting to that sting. So they knew that. Then they gave him the medicine, and then they said, so he sent me the data to see whether it is working or not. So they said, the report itself said that if you see the antibodies, that doesn't mean it is positive, because once the antibodies are made, they are made. They are not going to subside that soon. So you have to look for any other marker, right? So body is going to make the antibody. So when they give the vaccine, they look for the antibody in the body. Is the vaccine working? Has the antibody been made? Now, to answer your other question, what if the vaccine itself gives the disease, right? Because you're using the virus particle. That is your question, right?
The question is that you have vaccinated the patient.
Yeah.
So your intention is to see whether it is preventing patients from being infected by the virus.
Yeah,
okay?
so you will look for the antibodies against that virus. That is the way to find out. So usually when we get vaccination, nobody comes and tests us that whether we are going to get infected. Nobody tests. We have vaccination, we are supposed to be okay. But if somebody is really following up something, you can look for those antibodies, which is very, very easy to do it. Okay. So what you do is that you take the antigen on a chip or anything like a surface, glass surface, and you take the sera and you add it there. And the antibody will bind, of course, if the antigen is there. And then you add a fluorescent molecule which will bind any antibody. So then you wash it away. So only the antibody which is bound to the antigen will remain. Everything else will wash off. Then you add the secondary thing with the fluorescent thing, and you will get the signal if the antibody is there. So that is very easy to check that this person has responded. But you, I, all of us have gotten vaccination. Nobody has followed up on us, right? Whether we 99%, 100% of the time we have antibodies towards that. That is our body's immune response. Now, but I don't know if you asked or someone else asked, what if that the person gets vaccinated? So you don't give the live virus, you give like a tiny bit of piece of a protein kind of thing as an antigen. So there is no chance of, there is no virus there. It is just like a broken nail unique to the virus. So that's not going to cause any damage anyway, but it is going to trigger a strong immune response. So that's the beauty of the body. Just add the tiniest bit in the body. And that's how we usually get fever the next day when we get vaccinated and we are sore and things like that. That means the immune system is working. Yeah.
Does cancer reduces antibodies count also?
Why should it reduce antibodies count?
Because since antibodies have potential to kill them.
No, only if the antibodies are going to be produced against the tumor.
But in general, we have antibodies.
But those antibodies have nothing to do with the tumor, right? If we have memory antibodies or anything, they have nothing to do with the tumor.
But we have innate immunity, which consists of IgM, IgA, IgG.
I don't know if the general antibody counts go down. I don't know that. Have you guys heard of the antibodies count going down? I know immune system is suppressed across the board.
Way around, but that's the question.
I don't know that. I don't even know that. Yeah, search it. Yeah. If the antibodies in general go down, I would think that the body is so resilient, the memory antibodies will remain there.
Another question. Right.
By any chance, patients got recovered from the cancer, 100% recovered. Does memory cell hold the memory of, coming back from cancer? Is it?
Unless the antibodies were formed against the antigens.
They were involved.
If they were not involved, there is no memory there. But if antibodies were formed, then there will be memory. So if you are killing them by chemotherapy or something, they are not going to form an immune response there. Yeah, so then there will be no memory. Even if you are giving immunotherapy, there is no antibodies involved there. So it's only when B cells find antigens and then they form antibodies and they kill the tumor, then there will be memory there. Otherwise, there will be no memory there. So if you get HPV virus thing, then you are protected from that because you have an immune response. But in general, these therapies, chemotherapy, immunotherapy, they don't involve antibodies. Oncolytic virus, no antibodies. Oncolytic virus can trigger an antibody response and then it will be protected. Anytime tumor antigens trigger immune response, keep in mind, maybe, I don't know, it is possible that the tumor microenvironment is so immunosuppressive that oncolytic viruses are killing the tumors, but the immunosuppressive tumor environment is not letting the immune system launch an attack on tumors. So it depends from cancer to cancer, to stage to stage. So there are many factors involved there. And then there is this heterogeneity of tumors. So you are working with one antigen, but the other part of the tumor is not even expressing that antigen, right? So then they are not going to be affected by the vaccine. And the tumor may change shape. It may downregulate that antigen. It can downregulate the whole MSC1. So it can downregulate the antigen. This is very well known that if you recognize a new antigen, it will downregulate the new antigen. If you form something against a new antigen, it will downregulate that new antigen. It is like a virus constantly mutating. How is the COVID case right now in India? Is it like once in a while kind of thing?
I don't think they are testing for it.
They are not testing.
It's gone out of protocol because there was a time where they would recommend a COVID test if you had the symptoms, but I don't think doctors are doing it anymore. So I don't think they are large scale data on what the frequency is.
Right, it suddenly pops up like in this state the cases are rising, then it shut down.
Then people get immunity to that. That's the thing. Because it is not very bad, we gain immunity to that, which is good because self-immunity is good. Yeah. Yeah. But it is constantly evolving, and that's why I am getting alerts, there is a new COVID in rise, get vaccinated. So, I mean, in our area.
And can cancer be used as bioweapon as well?
Say that again?
Can cancer be used as bioweapon as well?
I don't know.
I mean,
It's a
ma'am,
very
a
detailed thing, no?
a nuke would be technically a bioweapon that is inducing cancer. So, I guess
Nuclear
nuke,
bomb. It
Ah.
will
a nuke would
induce
be a large
cancer.
scale cancer bomb essentially.
But it's in a micro level.
I mean, that would be very insidious if somebody managed to induce cancer with like very small level of impact that is not external.
Oncogenic viruses could be created, no?
There are
Oncogenic-
And they could be
virus.
carrier
Yeah.
also, no?
Yeah.
They are carriers
Yeah.
if they infect
be used
human
as-
cells.
Ah, they could potentially use
But
as
there are
bioweapon.
oncogenic like HPV is valid.
Oncogenic
but
virus.
the frequency of it causing cancer is pretty low. If I'm not
I don't know
Because
that.
there's a vaccine against it, and it's pretty curable.
I mean, it's fairly recent the vaccine
Yeah.
came out, but
But the thing is, we can create it in a lab. I mean, we can change something in that virus.
you
to
can obviously
it
make the
more,
perfect thing in the lab. Yes, that is true.
more oncogenic and use it as bioweapon.
Very devious.
Discussion going somewhere.
Very devious. We were supposed to cure cancer.
Yes. So, ma'am, what I was going to ask that on the basis of this,
Please, yeah.
so
On the basis of this neoantigen and antibody interaction, could there be a day when we can use similar tests as we use for viruses, the QRT-PCR kind of test for cancers?
You can even do it today. So, people do biopsy. Okay. So, for prostate, they do biopsy. For breast, they do biopsy. I'm sure they do it for liver also. Okay. So, biopsy is a must to do. Unless, we feed something like you were saying. Remind me your name again.
Tamal,
Kamal.
Tamal, TAML.
T-A-M? Tamal. Oh, so I never asked your name for sure. Okay, that's very nice name, Tamal. What does it mean?
Actually, I am from West Bengal, so there's a flower called
Tamal. Is it not Kamal?
No.
It is Tamal. Okay, so I am biased, so I heard Kamal. That's what your biasness is, right? Whatever you are used to hearing, you
Even outside, I only say Kamal.
people are biased. People are biased. What do you do, right? I am one of them. So by the time it traveled to my ears, it became Kamal. So Tamal, like you were saying that for brain cancer they give this special thing. So you don't want to go to the brain to do biopsies. It's much better to give a radioactive material. I mean, of course, low dose to scan the brain to see if that is overexpressed. So biopsy is pretty common to find out what exactly is wrong with that cancer. But remind me, what was your question again?
Ma'am, my question was the similar way we were doing for viruses, especially for COVID, we used QRT-PCR and we got the positive or negative result in less than 24 hours. So something like that for COVID. So we
No,
could
you can do
act
it.
early.
Yeah, you can do it. So suppose you have the tiniest and tiniest piece of DNA, right, from the biopsy. You obviously have to have something from the tumor cells. For COVID, you can do it from the mouth, but for that you have to go to the tumor cells, right? So if you go to the tumor cell, BRCA1, you can take a swab and see if BRCA1, BRCA2 is there, right? In like less than, same QRT-PCR will tell you whether it is there or not there. If it is a KRAS mutation, you can make the primer towards the mutant site and you will know right away. You can do sequencing. So everything can be done in 24 hours. Every single thing can be done in 24 hours. And you will know exactly what is wrong. And when you say this next generation sequencing, what they do, last time when I came to Gyan course, I actually taught that next generation sequencing also. So you can sequence the whole thing in no time basically without going into details. Okay, so that's what people are doing. If you get a tiny bit of biopsy, you can check the DNA, you can check the RNA, you can check the protein, you can check everything. And it's a very fast turnaround time. So you can do that. The only limitation is that if the tumor is deep inside the body, people are reluctant to do biopsy there. Like I know of that person with liver cancer, the doctor would not touch it because they are afraid that they will spread the tumor. So they are waiting for immunotherapy to kick in so that things are under control. And maybe it is possible that hepatocellular carcinoma doesn't have too many targets. So the more generic thing is more common. Like breast cancer, they will immediately go for targeted therapy. If there's something wrong with RAS, they will immediately go for RAS inhibitors. But the truth is that pancreatic cancer is very aggressive. 100 different things go wrong. You have to test it. You really have to test it. It could be KRAS. And if it is KRAS, you give KRAS. You get TBK1 inhibitor in combination. Oh my God, you're going to kill it completely. Right? And KRAS, the beauty is that it is mutated. So we have inhibitor which will only inhibit the mutated one. So targeted therapy is a must if it is possible. One should not go for chemotherapy. Have you guys heard of Alex Trebek? Anyone here? No. Have you heard of the programs called Jeopardy? Not heard. Jeopardy. It's a game show. It's been going on since 1970s in US. So it is like a trivia thing. You ask questions, and it is probably the most popular game in the world, right? So Alex Trebek was the host since the beginning, and he passed away a few years ago. He had pancreatic cancer. So he also said at once he was taking chemotherapy, and at the end he said, chemotherapy will kill me, cancer will not. So it is pretty bad effect, chemotherapy. He was, relatively, I think in his late 60s or early 70s, something like that. So chemotherapy is bad, but it has still cured many people. The person I was telling about, 40 years old, the doctor surgically removed lot of things from her mouth and still gave chemotherapy, and she's completely free of cancer. So it took her 1 year to recover, but
Ma'am, today you told us about that there are peoples working on the pH-dependent tags drug, pH-dependent tag drug. So, ma'am, will not it only based on the fact that the microbial environment of a tumor is acidic, so the drug will get active there and kill all the cells of that area. So, ma'am, won't it be triggered by the natural gut microbial habitat and the other natural microbial habitat our body?
So not microbial, microenvironmental, right? For tumor it is microenvironmental. So you are right, if it goes to the stomach it is too acidic. So maybe they are giving intravenously there. That's a good point. It cannot survive in the stomach then. Stomach is super acidic. Yeah, so it will be given intravenously. People are also doing a lot of fun stuff. Some things will reach the clinical trial, but you also have to do a lot of fun stuff. You don't know what is going to work at the end of the day.
But ma'am, if it is given intravenously too, the blood only, there are two organs in the body where blood doesn't reach the blood-brain barrier, so the brain and the testicular region. Otherwise, all the organs get the blood supply. So, stomach cell, the stomach cells which are creating the pH change, which are releasing the acids, they will have acidic environment.
So let me correct you. Blood definitely reaches the brain.
But
It is purified, no? Blood-brain barrier, so only.
No, blood is still going. Blood has nothing to do with blood-brain barrier. The compounds may not reach
Yeah,
cross the
ma'am,
blood-brain
that's
barrier.
what I meant to say.
Oh, that's what you meant to say. So
Not in the raw form. Blood is getting purified before entering the brain. That was I meant to say.
So here is the thing. So it depends on how big it is, right? So sometimes it is a good thing it doesn't cross blood-brain barrier, because you don't want everything to go to the brain unless it is targeting brain cancer, right? So that is good news. But I don't know if the tumor secreting cells, the cells which are secreting SCL will cleave it. I don't know that part. Or even if it is cleaving to some extent, maybe a lot will still reach the tumor site. See, this is not FDA approved. This is what people are doing in the lab. So this is what they are testing in the animals. And animals, when they grow tumors, usually they put it in the hand, like shoulders or thigh. So they form visible tumors, so they can give it intratumor, like just injection that way. Or even if you give it in a mouse, it's not a clinical trial, anything. So I don't know for pH linker. I have not seen FDA approved, but people are doing that. Go ahead, you were saying.
I didn't get the part of his question. What did you mean when you said that the cells that are producing acid will have an effect? I don't get it. Why would the blood be affected by that?
No, they are pH sensitive. So area around them will
I
have
don't-
low pH.
I think the stomach specifically would have the low pH, but the gut, the cell area having low pH makes no sense, because the other
The
cells
blood
would be
has
affected by
nothing to
it.
do with that.
Yeah.
Blood vessels will have nothing to do with that. So in our stomach, there is no blood floating around. Free blood is not there. It is in the vessels, right? So the stomach which is secreting the SCL, right? It is the stomach which has it, but there is no blood mixed in SCL there. Otherwise, we'll be throwing up blood every now and then.
Yes.
So blood is not there, right? Unless there is some serious bleeding there. But having said that, I have not seen any pH sensitive which is FDA approved. If it is, I am not aware of it. But people are making this kind of thing.
We're done too early.
Hmm?
We're done too early today.
That is good, right? That is good. You are the, the hard workers, the persisters continuously. And you will learn a lot also. The thing is that even if you forget that thing, if you have the background, you can always pick up any time because you have the background. Like I teach metabolism to students, right? And if you teach metabolism, there are 10 steps of glycolysis and 8 steps of Krebs cycle and this and that. Obviously, over time you will not remember. But if you know glycolysis, you can very easily connect to anything, and you can always check if you're reading about something. So just having the background is enough, because you will at least know what to search for when you are searching. If you don't have the background, even ChatGPT cannot help you, right? Because you don't even know what question to ask there, right? And also ChatGPT and Google Gemini are very you can manipulate them very easily, right? If you give them something, they will make a connection which does not exist.
They start making bluff out of it.
Huh?
They start making bluff out of it.
Yeah, they can actually make bluff out of it. That's the best way to put it. You put 3 things there and 3 things do not exist in literature, and they will make it for you.
Yeah.
And oftentimes Google Gemini and ChatGPT will give you different answers for the same question. Yeah, it is very scary.
So they have slowed down our learning process also.
Yeah. Actually, I wasn't aware of ChatGPT until very recently, because usually people Like students are the first one to know all the beautiful things. So what happened is that one of my students was applying to come to IIT Indore in Venkatesh's lab, right? So he did not come because he was an international student, and if you go out of US, you never know if you can come back again, right? So that's how at the last minute he said, if I go, I don't think I will come back again here, so let me stay here. So he didn't come, but he was applying for it, and so he wrote an essay, some essay, and I corrected it, right? And I thought it was a wonderful, and then he said, let me correct it, and it was even more beautiful. I said, wow, you write much better than me. I did not know ChatGPT wrote for him. But he was just very shy. He didn't tell me the secret, right? Then I was teaching a course and there were 2 TAs. So I said, it was always hard for me to make questions for the exam because you have to come up with innovative questions. And as you can see, I don't have much questions to ask you guys. So, right, I have to work hard to come up with questions. So I told the TAs, you guys make the question. And then one person said, use ChatGPT, it will make 100 questions for you in a second. And he showed me. That's when I realized, otherwise I would have not known about ChatGPT even today. And he had the subscription for that, so he can do anything. Yeah. We learned the hard way. But having said that, it is very useful because it can give you answers so quickly. It is faster than Google. Just searching directly in Google, right? And if you know enough, then you can see right from wrong also. So it is useful. So what are the questions we need to discuss that? Oncolytic virus, are they FDA approved? I have to check that.
Why are they not more used more?
Used more, that is one question I will check.
Do the antibody count go down?
Do antibodies count go down? That's a very good question. And what else? We all will check. And what else? So what is the situation in your country? People are getting less disease or more disease? Which one is more prevalent?
Mostly infectious diseases.
Infectious diseases. So cancer due to infection also?
Cancer cases low.
Less. Okay, that's good. In US, cancer is one of the big thing. That's why there is so much money devoted, right? Cancer is really good. Part of it could be like people are eating out all the time, right? And eating preservatives and tinned food. That could be one thing. And the biggest problem in all Western countries, including US, is Alzheimer's disease because people have much longer life, so the brain degenerates. And that is the biggest burden on society because these people live longer but very poor quality of life. So it's a very, very poor quality because they don't recognize anyone. So you hope that the family is very supportive, otherwise it's just really bad.
Ma'am, does brain degeneration occurs due to oxidative? Does it have
Oxidative stress is the root cause for that.
Absolutely right.
Yeah.
But we have antidote to it. We do meditation and yoga.
Meditation is a known thing that it prevents a lot of diseases. Meditation is really magical, honestly magical. Yeah. You do meditation. That's very good. All of us need that. Honestly, all of us need that. And we never have an excuse, it is no time, it's just your rest of the day goes so well after that. Yeah, that's very good. I like that you are using so many colors. It is reminding me of my own days.
I want to keep this notebook for a reference throughout for my career maybe.
That is a very good thing, yeah. We'll talk after the record.
Ma'am, I had asked you the question about the DNA being viral DNA possibly being mutated due to it being replicating inside the tumor cells. I think I might have found the answer. I just want to confirm with you.
Yeah, absolutely.
I remember that the viral DNA itself the replication process is very fast and it is very short-lived. So could it be possible that the ROS damage just does not get it to in time, so that the DNA would be changed that as much?
So I don't think for viruses ROS is a problem. Okay, I don't think so. It is for us ROS is a problem. ROS is not like immediate kind of thing unless you add H2O2 on something, right? So viruses are rapidly dividing. So in their lifetime, they're not even exposed to the, for us, like our brain gets ROS and it is there for like years and years. That's when the damage accumulates. If somebody pours H2O2 on virus, then it is there. But I don't think oxidative stress affects these guys that much. Same thing with E. coli. If you pour H2O2 on that, that's when it is doing. People who live longer, like dogs or cats or us, right? They are exposed to oxidative stress induced damage because, yeah, it is not applicable there. See, this is in my opinion, I don't know 100%, but in my opinion, we have a lot less Alzheimer's disease here. And it is partly due to the food that we eat. So turmeric is antioxidant, right? And usually, oftentimes we make fresh food, right? We don't use preservatives. So all of these things add up. In Kenya, do you have cases of Alzheimer's disease? Have you heard of it? You have not heard of it, right? So, see, people, different food habits. In US, cancer is so prevalent. Every second person you hear with cancer. And you know, air is so clean. Air is so clean, no pollution, no infection. You have annual checkup. You catch the disease before it comes. So there are so many good things happening there. Even then, cancer is extremely prevalent. Part of it, people are stressed all the time. Yeah.